Hello, I was just checking that everything is fine. Hello, can you hear me?
Yes. Who is this? Sorry.
This is Sanjay Singh.
Oh, hi. Good morning. Welcome. So yes, you are audible.
Yes,
if you want, you can check your slides to see if you can show.
Yeah, I'm going to check my slides.
Okay.
In a minute time. Okay. Thank you.
Good morning, ma'am. Hello, ma'am.
There's a lot of echo. Can you repeat again? Can you say something?
Yes.
Hello?
Yes. Hello.
It's
Yes,
fine
ma'am.
now. I think there were two devices connected. That is.
Okay.
Okay. Now it's okay.
Hello, ma'am. Hello, Dr. Sanjay. He was there.
Hello. Message. He is logged in. Hi.
Okay,
Okay.
Can you hear me? This is Sanjay.
Hello,
Yeah,
sir.
hello.
Yes, sir. Your voice is clear.
Okay, so I'm just sharing my screen now.
Okay, sir.
Good morning, Dr. Sanjay.
Good morning.
Okay. So, good morning to all of you. Dr. Swasti ma'am is here. Ma'am. Yes,
Good morning. So should we start? I think it's 10 already, so we can start.
Yes, ma'am.
Good morning, everyone. and very warm welcome to this 2-day online event. And on behalf of the Department of Molecular Medicine and Biotechnology, I welcome all the participants and the registrants who have joined us from SGPGI and also from various other institutes. So this is actually going to be a very relevant topic, and as you can see in the flyer, we have an incredible panel of speakers who will be enlightening us about the immune cell activation and its regulation in various diseases. So as you can see that today's talk is basically restricted to the COVID-19 disease, while tomorrow some of the non-COVID diseases will be covered. And I'm sure that we all will be benefited, both basic and clinical researchers will be benefited to enhance our understanding about the critical aspects of immune cell regulation advancements and the tools which are available for better disease outcome. So without taking much time, I think we should begin the session, but I like to encourage all the participants to please use their chat box for any queries they have. And once again, I like to thank our departmental faculty, Dr. Alok and Dr. Santosh, who has actually put in a lot of effort and come up with this relevant topic and incredible list of speakers. So I welcome all the speakers once again and request Dr. Alok to please begin the session by introducing our keynote speaker, Dr. Sanjay. Over to you, Dr. Alok.
Thank you, ma'am. Good morning to all of you. So Dr. Sanjay is the Chief Executive Officer of Genova Biopharmaceutical Limited, Pune, India, since 2006.
Recording in progress.
Sorry. Yeah. Technology development and ACS affordable. I'm sorry, so there is a glitch for some internet. So Genova Biopharmaceutical Limited is a biotechnology company dedicated to the development and commercialization of safe, efficacious, and affordable biotherapeutics. And Dr. Singh focused on the innovation in manufacturing technology that culminate in the commercialization of 7 life-saving biotherapeutics in the cardiovascular, neurology, nephrology, and oncology market. Under his leadership, his team pioneered the work that led to its approval of Tenet Plus for acute ischemic stroke, the first time globally secure global patent. The Department of Biotechnology recognized his innovation for Biotech Product Process Development and Commercialization Award 2019, and it found its way in the list of drugs for emergency care for stroke management in the guidance and prevention and management of stroke issued by Ministry of Health and Family Welfare, Government of India. Under him, Genova has directed their research to cater to vaccine development and related disease, in particularly malaria, HPV, leishmania, and tuberculosis, in partnership with various national and global organizations, including the NIH, US, FDA. Dr. Singh is currently a member of various important committees of national relevance, which notable include the Scientific Advisory Board of the National Center for Science, Pune, Research Council of IITR Lucknow, National Committee for Biotechnology, Confederation of Indian Industry, New Delhi. Additionally, he is an invited member of Indo-US Vaccine Action Program, which is the WAP, and contributing towards the national interest. Dr. Singh served as a member of committee for the development of India guidelines on similar biologics constituted by DBT, and Central Drug Standard Control Organizations, which is the CDSCO, and the Ministry of Health and Family Welfare. Contribution of this committee led to establishment of a transparent path for successful development and approval process to market the biosimilar drug product in India. Dr. Singh holds a PhD degree in biochemistry from CDRI Lucknow. And before joining Genova, he headed the antigen research section at the malaria vaccine development unit of National Institute of Allergy and Infectious Disease, NIH, USA. With this brief introduction, I would like to extend warm welcome to Dr. Singh. Please, sir, over to you.
Yeah, Santosh, can you see? You can see my first slide.
Yes, sir. We can.
Yes, sir. We can see that.
Okay, thank you very much. Go ahead. So can I start my presentation?
Yes, sir. Yes, sir.
Okay. So thank you very much for inviting me for this talk. I would like to share my few memories of SGPGI when I was doing PhD somewhere in 1990 and 1991. So the latest journal used to come in SGPGI. So I used to drive from CDRI to SGPGI, spend half a day, eat lunch in the canteen and come back in the evening. So every Saturday, Sunday, I used to go to SGPGI. So these are my fond memories from the SGPGI. And thanks, Alok is there. Alok has joined, and I'm also part of the advisory council of the institute. So I'm very happy to participate here, and I'm giving a talk in such a sense that it should be a little bit kind of general audience also, but also gives a just what the topic is, which is immunomodulation. So a new era has come up in the last couple of years, which is mRNA-based vaccines. And where Genova is focusing on two types of vaccines overall. One is preventive vaccine, which is one of the examples today we are going to give, is COVID-19. And second is the therapeutic vaccine. And here we started a long time back, like 3 years before we started this program. And we have a whole vaccine portfolio, but mRNA, we started somewhere 3 years back and looking into it, that what advantages gives. So before going that what mRNA is going to possibility give advantage, I would like to discuss overall what are the vaccine platform and why this new immune activator has come into the picture. So first one is vaccine 1.0. And this was initially developed, this platform, by Professor Jenner at Oxford, 1900th century, that where you can take the whole virus or whole organism and one way or other you attenuate it and use as a whole organism to use as a vaccine or immunomodulator. And here the examples are hepatitis, polio, rabies, influenza, all these kind of things you can have as an advantage for this. Then the second generation platform started. And the second generation platform started, it came with the recombinant proteins kind of thing, which is, you can say, much more defined from the first generation platform. And here we know the sequence of the protein, we know the genetic sequence of the protein, recombinantly you can express, you can fold it, you can characterize it, and come to the close the native structure of the protein, and that is used for immunization to get the immune effect into the body for productive. So here the best example was hepatitis B vaccine. It is produced in Saccharomyces or recombinantly or produced in Pichia. So this is the second platform came. So a big movement has happened from first to second. In the first platform, you have to make big facilities, which may be, if something like polio, it has to be contained. It is BSL-3 or BSL-4 facility or rabies facility. So that risk factor lowered down in the generation 2. Here it is first thing more defined, and you are giving those particular components which are required as a vaccine candidate. Because in whole organism, not everything is required to do its job. Then the third platform started. This started a couple of years back, maybe 20 years back. And here people started doing DNA or adenovirus-based vaccination. In this, other than protein, now you have made a DNA construct or adenovirus construct and you have given to the body, and let's body makes the mRNA, the body makes the protein, and then it is close to the native structure because it has been made in the system. And here, one of the emergency authorization was given for Ebola vaccine a couple of years back by the US FDA, and the second is the AstraZeneca vaccine, which is under emergency authorization of use. It has been approved in our country. It is in great use. And the last platform comes, which is mRNA-based vaccine, and where we comes into the picture also one of the players, but already 2 vaccines have been given emergency authorization, Moderna and Pfizer. Here we have moved one more step closer to this. So we have given the mRNA signal to the body, and body has made its protein, and it is closest to near one. Again, manufacturing and safety, which I said in the generation 1, you have to have these kind of big facilities. In generation 2, you have to make a synthetic gene, you have to make a recombinant cell line, you have to characterize the cell line, then you have to go for big bioreactors, and from big bioreactors, you have to produce it and characterize and all that. Generation 3 has given advantage that here you can take HEK cells or BHK cells and produce the plasmid and purify the plasmid you have given. In the last one, it is totally synthetic. And when I will walk through the process, I will tell you, here you have just a DNA plasmid, and DNA plasmid you can take in a tube and just add different enzymes at different processes, and your mRNA is prepared and you just purify it. So this ease of manufacturing was the one, and then the safety and efficacy has been now proven. So this platform is going to be future of the vaccinology in time to come. So this is the background for it. So if we see what I have said, again, I'm to reiterate just to give the audience, this is the vaccine 1, this is the vaccine 2, this is the vaccine 3, and this is the last is the vaccine 4. So in vaccine 4, we just need in vitro transcription, cap it from the 5' in, verify it single step or 2 step. And why mRNA was not in the field, though around 10 to 12 years before investment has started going into this, because delivery was a problem, and then you fill and finish and you can release it. In all 3 platforms, it will take around 45 days or 50 days to make, but mRNA vaccine, you can start in the morning, in the evening it is made. You can fill and finish, and then you can do all the tests to release it. So this is one of the, another ease has been given. So economics of the process, economics of the manufacturing and time to manufacturing, these are the things which has made it more attractive. Now, I don't want to go into detail, but there are so many, this is the last update where we have hundreds of vaccines are there, which is protein subunit vaccine, DNA vaccines, inactivated virus vaccine, and RNA vaccine. 18 RNA vaccine at different stages of trial. So we are also one of them in the world. Now I'm going further detail into this processes, and we have created a self-amplifying mRNA. Self-amplifying mRNA, either you give one mRNA or you give a whole vehicle by which multiple copies of mRNA can be made. And then the delivery system, it's nanolipid. So you use that mRNA and you coat it on, or you entrap into the nanolipid carrier, because otherwise mRNA is very labile, it will be destroyed. We started this program somewhere in last May, June with the DBT funding. They have given us some seed funding of 25 crores. So I'm very thankful DBT, they thought about it and they invested boldly into it. But what kind of thought process we were having, that we will take one of the spike protein, at that time it was D614G was the most prominent, because from Wuhan, which the virus was started from Wuhan, it mutated itself worldwide. Self-amplifying, so possibly we don't have to give many copies of mRNA. You give one copy, the rest of the copies are made in the body, and its time is around 8 to 20 days. It's self-destroyed, and it is in the cytoplasm only. It doesn't cross the cytoplasm to nucleus site, so there is no chance of its integration. And this absorption chemistry we developed with our partners back in collaborators in US. So this is a nanolipid carrier. the biggest challenge we took, can we make it 2 to 8 degrees centigrade stable? Because a vaccine in India, if we make at -70, it is almost unaffordable. And the other part was, can it be given to the COVID recovered people? Because COVID, different level of infections are there, but they are not, even after COVID, maybe 3 to 4 months, they are protected, but again, they are going to be prone to it. And Alok knows, one of our scientists, Dr. Rajesh Singh, he got twice infected. So we have around 6 people in the company who got twice infected, once infected, and after 3 to 4 months again got infected. And then potentially data can be developed for administration with other approved vaccines, because if the ease of vaccination is so easy for manufacturing this. So we were given a second round of funding. Once the proof of concept we established, it was reviewed by multi-layer and multi-time committee, and we were funded another 100 crores for scale up and CT. Why I'm putting these numbers? Because I just want to say, and this is for the young scientists, now government and funding agencies are very, very open for big funding if the concept and science is innovative enough. So this is the vaccine vial, which is in the trial at present. So now going further into it, so this is basically the construct you see. It has an SP1, SP2, and SP3, and SP4. These are the machinery for it. But basically, this is the SARS-CoV-2 spike protein, followed by UTR at the 3' end. Once we, by in vitro translation, we make self-amplifying mRNA. We just take a nanolipid carrier and put on that simple mixing, just 2 mix, and it is positive and negative charge, but dispersity and many things get followed into it, and the vaccine is made. And we have termed our vaccine HGCO19. If you see from a data point of view, this is basically the mRNA. So when we made this mRNA, it was 12 kilobase plus, and the ladder which was available worldwide was only 9 kilobases. So this is the mRNA, and we have developed method by reverse pages, as you can see. A very beautiful peak comes up mRNA at 260. Now, once we mix these two things, what the figure, if you see in the last, what we said, mRNA and this nanolipid is mixed, then this is the kind of size it comes out. So if you see the agarose gel, which is given, it is lane 2, it is the mRNA, free mRNA, but when we mix it, the lane 4 and 5, that mRNA goes away, but we can extract it back. And if you can see lane 6 and lane 7. The most important thing is lane 3. If you take this mRNA and treat with RNAs, mRNA goes away. But when we have made a particle of this mRNA, it is protected, which is the last 7 lane. So this become a particle, which is average beta size is around 89 nanometers. PDI is X, and this is the basically complex we make. Now, what we did, we have done more than 30 experiments in mouse, rabbit, hamster, rats, monkeys, all that kind of thing. But when the proof of concept was done, then I'm going to present first set of data to you. When we produce the cGMP batches, before going into human, we tested it again, because many times what has happened, what results you get in the lab, and when you convert into industrial process, and then you test it, it has to be fully tested that similar kind of result has been proven. So here we take a pre-bleed from the mouse, then give first dose at day 0, as given in the cartoon. We withdraw the blood at day 14, and then wait day 28, we again withdraw the blood, and day 29, we give it the booster, and blood is connected on day 57. So what is the humoral response? So if in panel A, you see downsize on day 0 and day 29 and day 57, there is a immune response which get boosted when we give the first boost at day 29, and the titers are in skyrocketing. But the next question comes that this is a virus neutralization titer. So virus neutralization titer is done by one of the institute here in Pune. It is again DBT supported institute with IRSA, and they have done the virus neutralization assay because we don't have BSL-3 facilities. So when we tested this, our day 57 sera, it is around 800 to 1,000-fold lower. You dilute to 800 to 1,000-fold dilution, it gives 50% inhibition. So this data was very, very encouraging for us as the research product was showing, the same CGMP product is showing. And there is a correlation between antibody titer and basically the PRINT50 titer. And some of the mouse shows even this neutralization titer of 2,564. Now, how it looks, because doing print assay is a very long and tedious journey. But here, if you see day 14, day 28, day 42, again, we have put it together. On left side is the in log fold the antibody titer, but in the right hand side you can see it is the surrogate virus neutralization assay. And this assay is a very unique assay. It has been developed between the University of Duke and Singapore University collaboration. And now it is available as a commercial kit. And it was also published in Nature Biotechnology in last year. And this gives a very good, it gives very good results, which you can compare with the convalescent people. So if the last panel, which I have given to you, on day one, on number one, I myself, when I was not infected, it was 0. Then second one who worked on the mRNA, Ajay. And the third one, this person came back from the ventilator. So its blood titers were neutralizing titer were around 60% when we had used the surrogate virus neutralization assay. The fourth person was in the hospital for 15-20 days. The fifth person was very mild, but the sixth one is again said Dr. Rajesh Singh blood, which is convalescent, and he was twice infected. So basically what we say, the neutralization titer are between 30 to 60% in case of people who has gone infected and recovered. But if you see in our mouse data, we are getting on the same assay the neutralization titer around 90%. So we have created the antibody titer, which is much more higher as compared to convalescent sera. And this, note this factor because this is, we are going to connect it somewhere later. Next one. Now, from the immunology point of what is looks like both immune modulations are going to make a difference. One is humoral and one is cellular. So in cellular responses, if you see the panel D, here we have taken the total dendritic cells and you can see, yes, some stimulation is there. But when we use activated dendritic cell gating by CD80, CD86, then you can see there is a statistical significance we have got. In the same way, when we take germ center B cells using CD19 gate and FASDL7 cells, this is so important that there is a uproot is saying, but when we take a spike-specific germ cell B cells, then there is a statistical significance. This was one of our most important data till date. We found that we can induce the germ cells against this. So memory is there and it will come back. So this paper is under review at present and hopefully we will get accepted very soon. So this is a part of I wanted to give humoral response and cellular response, what we are getting, but now we have tons of data on this. We have taken on different mutant and all kind of things are going on. So now we have gone into the phase 1 and we have completed phase 1 trial. 82 people have been completed. And here what we use, one arm was the placebo arm and the second arm was 5 microgram, either 10 microgram or 25 microgram. We wanted to do in 2 cohort, 18 to 55 year group, 60 people and 56 to 70, another cohort. But when we moved into cohort 2, we were getting around 86% people who are seropositive one way or other. So our down rate was very bad. So we only considered 22 people into that. So we can complete 5, 10 microgram, 25. So we have given on day 0, and then we have reviewed them on day 8. The second vaccination is on day 29. Same, either prime boost or placebo has been given. Follow-up, we have done 36 and 57, and interim analysis is going on. As present, we cannot present the data because this has to be first presented to the Data Safety Monitoring Board. But we have great safety, reactogenicity, tolerability, immunogenicity, and recruitment completed. There is not a single serious adverse event we have found. Some local adverse event like mild grade 1, some pain in the hand, and moderate grade 2, somebody had a one-day fever but was self-resolving. So one set of DSMB has done, but the final set is DSMB is due on 10th of August. Once it is done, then we will submit the data to DCGI. So at present, we are not presenting the data, but data is encouraging. Now coming back on why it is very, very critical, because we have to one way or stop this pandemic. If pandemic is not stopping, we are in a difficult situation in terms of economic policy, which drive the country, but in terms of all operations. So here, what it takes to this, I'm changing the topic, but it is very, very important for the group to understand what we are changing it. If we have to start the pandemic, this came somewhere in last September itself when none of the vaccine was approved. And they said that vaccine efficacy versus herd immunity that make a sense. So if we have a population of 138 people, roughly we said there was a time, maybe we said we have 20% herd immunity, but somehow we were having a lot of herd immunity in Mumbai and Delhi before second wave, but these were the most affected cities. So if we have a mRNA vaccine, which has shown around 95% efficacy, we might need in combination of two only 40% coverage, so 108 doses. But if we have a vaccine efficacy which is 70%, we will need 150 crore doses. But if we have the vaccine which is 50% efficacious, so we have to inoculate around 208 crore doses. And this is prime boost strategy, but at present, we do not know how it will go, how long the vaccine, how the antibodies are staying, and how the next level of infection people are safe with that. So here I'm changing the topic, and something has been published a couple of weeks back. They have demonstrated, this is example for AstraZeneca vaccine and Pfizer-BioNTech vaccine. So Pfizer-BioNTech in blue and AstraZeneca is in pink color, you can see. So amazing, we are seeing that after 21 or 41 days, by the time we reach to 70 days, the titers are waning. This is quite unusual things. So titers are going down. And if we see another publication which came around 20th of July, here again they did mRNA vaccine, BioNTech vaccine. And here if you see something more to see, that on day 0 is this two type of people. One is seronegative and one is seropositive. Seropositive means people who one way or other already infected or something has been given to premier vaccine has been given to them. If the titer, if you see in both the cases, when you give the dose of the vaccine, it goes up, but still it wanes in case of seropositive also, in case of seronegative also. So titers are waning down. What kind of effect is going to happen, we do not know. But at present, what we see, titers are waning down. This is another paper has been published, I think last week again. This is a group, same paper that they have presented in some other format. And this is very interesting to see. The blue one is the people who are seronegative, they have been given 2 doses. But red one, the pink color is, these are the people who are seropositive. So their titer goes very, very high. So at present looks like whole country population, around 70% people or 75% people are seropositive. Because when we were enrolling the people, volunteers for our phase 1 trial, we found more than 75% people were not getting enrolled because they were having seropositivity, one level or other level. So there is some memory is there from them. Now, this is another group has published paper on 23rd of July. Here, another very, very important factor, if you find that single dose in seropositivity, take it to the top. So which is, you can see from the color, kind of yellowish brown. But people who are seronegative, if you give them 2 doses, they reach to that particular titer. So if the country is having such a seropositive population, then possibly mRNA will do wonder with the 1 dose. This is the thought process at present going on. The data in our case will show it how we have gone when we will, enroll the seropositive and seronegative people both. So another part, if the cartoon, the same paper, the cartoon, if you see, you have given single dose to seropositive pre-existing immunity, and it take to the same level. So second dose is not making any difference in case of mRNA. But the first case where the new people are there, then you have to give 2 doses, and similar kind of data we are seeing. So hopefully that we may have some advantage when the vaccine will be in the market. Now, this is a very, very important factor if we culminate everything and want to say in similar fashion in interest how it is going to affect the data in India. So the blue set of people are seronegative and red are the seropositive. And if you see in the B panel, if you give single dose, the titers and cellular immunity, both goes up. But in seronegative, you have to give 2 doses, booster doses, then it touches to that particular point. So post-dose response and recall response, it is very high. So somehow mRNA has this ability because it is made in your own body, the way virus protein has been made, exactly mimic, it has a huge difference. On the right-hand panel side, I don't want to go into detail, but what I said that how the convalescent people make a difference. So convalescent people is one. If you see the convalescent sera, this came in Nature Medicine a couple of months back. And here, left side is the different vaccine has been given. So if you reach to convalescent side, what is related to the protectivity? So mRNA, both the vaccine has gone to the right side of the convalescent. So who looks like this is what they are giving them the highest efficacy as compared to different platforms of the vaccine has been used, either the protein vaccine or other vaccines have been used into this. Now coming back to next point, which is the variant of concern. So now there are a couple of variants of concern has been demonstrated, alpha, beta, gamma, and delta. So in all cases, is neutralizing activity or memory T cells are there, protective some immunity is there. So in clinical correlates protection are known, or we are trying to put clinical correlates protection, but if we give the mRNA vaccine, if recall comes, then we may have a huge advantage as compared to other platforms. This data has been said by others, and we are also going to have this kind of data available in next 1 or 2 months. Now, this is very, very important when I was called by the SGPGI to give a talk, because I want to encourage the young scientists to look into this, because mRNA platform looks like to be a disease agnostic platform. What that means, disease agnostic platform? So these are the only component. You need a lipid carrier, which is, we say this, the new lipid carrier which we have made, we say GNE, it is nano emulsion. Then you need a couple of recombinant enzymes. Then you need some cofactor and nucleotides and lipids. So you can create a stockpile for it. And any plasmid you want to take, and it is disease agnostic, whether you want to take for malaria or you want to take for tuberculosis or you want to take different mutant of COVID vaccine. We have made vaccine against all mutant. You just take that synthetic plasmid is not a problem. And now we have one of the very modern system codex. So here, it in the night you feed it, in 24 hours it make it and make the plasmid. You can add it here, you mix it, and the vaccine is ready. So what right side is giving, this is a very, very rapid platform. Once it is established, then this is a very, very rapid platform. Within 50 days, you can go into and you can inject it. Once your platform is approved. So if you ask why Genoa is late, because when we started this platform, we went to the RCGM, we presented all the data, we went for preclinical toxicity, and though the BioNTech and Pfizer were not subjected to animal preclinical toxicity, we did one preclinical toxicity in US. They brought out some rule which was introduced in 2019. They said you have to go for 2 preclinical toxicity. We did 2 preclinical toxicities, took us around 8 months, and then we started the clinical trial. But now platform is through. Hopefully we will get in time to come the approval, then mutant versions and other vaccine will not be a problem. So what I want to change the gear from him here and why we started, this is the part of the preventive vaccine, but therapeutic vaccine, why we started, I just want for the group to get, think about it, what is great this value will create. So we are working in the neoantigen therapy for cancer. So neoantigens are there. These are the aberrations of your own system. And somehow the CAR-T technology came. So one of our friends, they are at Tata Memorial Hospital, they want to create this CAR-T therapy. I don't want to go into detail of CAR T therapy, but it is basically the whole very costly affair to do the CAR T therapy. It in US, which is approved by CAR T therapy, it is around $500,000. I do not know how it will cost in India and how it will make, but the possibility is you can exploit the immune system and you can exploit your technology to make the vaccine. Can you combine both? So here is, if you have a tumor, you can pull it out and you can look for predictive algorithm of MHC binding capabilities in terms of neonatals and put in the same platform, and you can go ahead and inject in and see how the response is going to come. Same thing you can take from PBMCs, you can figure it out. So neoantigen is the new area where we are working together at present for last 2.5 years. And let's see how this field is going, but this is one of the very exciting one from where we started, but at present we ended at COVID, but this is the area where we are quite busy today. So thank you very much with this. So I think I have tried to cover my talk in well before time, and I want to open the discussion so question can be asked.
Thank you, Dr. Singh. It's a very informative and very nice presentation, and we look forward for this important mRNA vaccine development. And it's very exciting data for the remodeling of immune cells like B cells, T cells after mRNA therapy. So these are very interesting aspects for the vaccine development. Yeah, we know that there are lots of complex and there are lots of systems involved in the vaccine development, gender differences, age dependent differences. So, so there is one good question means what are the therapeutic windows means we can play here for the immune or humoral or cellular responses regarding the adjuvant development. So, is any comment on that?
So the nanolipid carrier which we have adjuvant squalene is there basically. So any adjuvant, if you want, which can be created on nanoemulsion platform, you can add these adjuvants. So we are testing different adjuvants. We are testing MPL, we have tested GLA, we have tested CPGs. So those things we are putting it and we are looking into it, that how, because this is not the only vaccine, we are working on different vaccines. So there is a huge opportunity. You want to screw in such a fashion where it can be helpful, but the data will tell what will be the most optimum. But for COVID vaccine, we have found the present system, squalene is there, it is good enough, it is doing its job.
Thank you. So I would like to request from the participant, if they have question, please write on the chat box and also raise the hand so I can ask. Okay, so Dr. Santosh.
Thank you, Dr. Sanjay, for the illuminating talk. It was really exciting to go through all the journey with the vaccine. Genova has taken initiative. So we know, like say, now we are going to have a flavor of a diverse vaccine, protein-based vaccines, mRNA vaccines, and Genova. Oh, okay. I'm sorry.
Go ahead. Santosh, you are on the mute.
Yeah,
yes.
We can hear you now.
Yeah. So since we already have the key players like Moderna and Pfizer, so what's the key difference in terms of like say the mRNA formulation, vaccine formulation? It's going to have like say for the Genova, so that the humoral responses and the CMA responses is going to be better or something. Can you comment something on that with in relation to the existing mRNA vaccines for the COVID?
So basically, the platform for us and Moderna and Pfizer are same, but there are some subtle differences. The first subtle difference is that they are using simple mRNA. So their doses are higher, maybe 100 micrograms for Moderna and around 30 micrograms or 35 micrograms for Pfizer BioNTech. We are using self-amplifying mRNA, so our doses hopefully to be much lower, like I have tested 5 10, 25, even my highest dose is not the what is their dose. So there is a huge dose sparing capacity should come up. The second thing, and which I showed to you, humoral and cellular responses, they are exactly same or even few percent better than them because of our self-amplifying. So it is very slowly expressing the mRNA in the system, and it is intriguing the or hitting the immune system slow. It is not just a bomb blast. So that may give us, as I showed to you, that germ cell activation. The second advantage which we have, their mRNA is entrapped in the nanolipid, so the manufacturing of that entrapment is very, very costly, and also difficulty to clarify it because whatsoever left over. We are using a different technology. We are wrapping on the surface based on positive and negative charge, and that has given some stability. But hopefully, very soon you will see that we will declare that we have made this vaccine which is stable at 25 degrees centigrade. So 25 degrees centigrade in Western term is the room temperature, but ourselves, we will say to 8 degrees centigrade is stable. We have the 6 months stability data today. Because you see in Jaisalmer, it will be 55 degrees centigrade and Srinagar will be 5 degrees centigrade. So there is no room temperature kind of thing. So hopefully, this is another most important because deployability of this vaccine is very, very important. If this vaccine makes such a high efficacy, we cannot create the whole infrastructure for -70, and we all are scientists creating the infrastructure for -70, taking it out from one refrigerator, keeping it 2 days for 2 to 8, and then taking out the vaccine and all that kind of. So this is hopefully Genoa innovation into this field, and this is the differentiation as compared to existing 2 vaccines.
So thank you. So in terms of the logistic to reach the the remote accessibility, so would we expect, like, say, the cost is also going to be going down with because that's the one thing, because people will have a choice for the protein based and mRNA, and definitely the stability is the one which Genova is going to have an added advantage. So, but in terms of the cost, how those logistics, I know, like, it's too early to ask, but yeah, I think. What's your opinion?
Sometime back, this question was asked to me from some of the government officials, what is the cost of this mRNA vaccine? So I said in another way, what is the cost of stopping this pandemic? Is this vaccine is equally costing, which can, if we stop this pandemic, because this is mRNA platform is least controversial and most effective vaccine proven worldwide. Only thing, the logistic was a problem, and some other things, because they will use it in their own countries first. They will fill the quota and then they will give to the rest of the world. But if now variants are coming, booster is coming, then these vaccine will not go outside their typical Western hemisphere. So now we have lost around last financial year around $1.3 trillion economy we have lost. We were around $4 trillion. We lost around huge amount. So is the cost of this vaccine should be $1.3 trillion? No, it is 1,000 times less. Now, again, still we are suffering, and we have gone the second wave, and how difficult second wave was for all over the country. So what is the cost of vaccine? At present, we are negotiating with cost of the vaccine, but certainly we are going to be very, very competitive to the existing vaccine, without a doubt. But yes, there is a difference between different vaccines, the manufacturing, the efficacy, the safety profile, and the scalability. So that defines a huge proportion, but we will be competitive without a doubt. And I come from where you are sitting in Lucknow, the neighboring district, Barabanki, and I will ensure that it reached my block. That was the basic motivation to develop 2 to 8 degree or 25 degrees stable, this most advanced vaccine.
And one last question, related to a CMR response study. So is there any contribution for the dendritic cells in addition to the T cells? So
possibly, at present we do not know, but when we are going to do the big phase 3 trials, then we have to see that how the people behave, because when we have done this trial, one of the person got infected in 5 microgram doses. And how we came to know, because 7 people of the family were infected, and 3 were in the hospital. So when the quarantine people came and they checked it, and they said whether this person is, so this person was totally asymptomatic, but he was infected, and within like 10 days, the infection got resolved. So we do not know at present what is, because this all factor is going to immunology, very complex subject, how it is safe and how long the protection lasts. And if the infection happen, how the infection creates symptomatic or asymptomatic or mild or moderate. Those factors are going time to come to differentiate it. But we know the correlates of protection are basically humoral, but cellular are also going to make a huge difference. So another speaker, Sasank, is going to do, I think Sasank Tripathi is going to do our challenge studies. So most of the question he is going to answer very soon.
Thank you. Thank you. Thank you very much. Yeah.
Thank you. Thank you, sir. So there is one question regarding from the Shashank Tripathi, means how do you avoid interferon activation by mRNA vaccine, which can be deleterious? Is any comment on that?
Yeah, so we have, so basically most of the type, this comes in terms of if you have double-stranded mRNA. So we have tried to verify that. So we have 0 double-stranded mRNA. And then our mRNA is going to sustain for a very long period of time, between 8 to 20 days. So even if some interferon activation has come, it is going to be cut down, and by that it will survive. And that was one of the reasons having self-amplifying mRNA. Yes, some strategies have been used where they have used kind of pseudo nucleotides they have used to convert it. But when we compared both the technologies before going into human trial, we found that self-amplifying was doing a better job.
So, okay, so Dr. Lokendra, yes, please go ahead.
Yeah. Am I audible?
Yes.
Yeah. So thank you, Dr. Sanjay. It was a really very nice, informative talk in today's time. And really, we are thankful that you are working on that direction. I have just a couple of quick questions. One thing is that since you mentioned that, in terms of other vaccines which are available, the waning down of the titer is very common, which is, suggesting that it will, slightly lower than as the month passes. So in that context, just the basic question. So even if it is the viral titer which is going down, if you challenge, maybe if it has cellular immunity or cellular response, it will go on again, right? So in that context, what you suggest that these vaccines which are already available in market, definitely the rate of hospitalization is very less. So what is your view on, in terms of your vaccine? Whether it will be used as a booster dose or maybe as a supplement for those which have already got vaccinated, and if their titer is going down, maybe it will help to retain that, or what kind of views you have on this perspective?
So there are two scenarios as present. I will say from the philosophical way, and then I will come especially to vaccine which we are developing. One is we have a population of around 138 crores, and this was the last census, but you can consider 140, 145 crores. By till yesterday, we have given around 48 or 49 crore doses. So still, if we have to immunize 100 crore, 200 crore doses are required. And for 200 crore doses, still we are short of 150 crore doses. This is the part one. So we need all players, lot of vaccines to participate and fulfill the needs so we can come back to normal. Now, the second question is yours, that what vaccine will do. Yes, these questions we are asking in our phase 3 trials. We may do some small studies where we will ask this question, just like if somebody is seropositive, we have given single dose and how it has done. But AstraZeneca vaccine was taken by the Angela Merkel, the Prime Minister of Germany, but she took the booster dose from mRNA. So that is possibility is there. This kind of possibilities are very open. And government is looking very seriously on this and helping us to design this clinical trial to answer these kind of questions. But we have to develop the data, and then we can say that it is making the difference with our vaccine.
So how long time do you expect that your vaccine is ready after? I think it's a first, it's in the first phase. So maybe second and third phase, it will take
So
around like
first phase we have completed. Hopefully next week we may go for phase second and phase 3 trial approval. Because another problem which has come, because you cannot keep placebo today, because it is unethical. You keep somebody placebo and they got infected and they suffer. So placebo is ethically is rejected especially when 2 vaccines are available. So we might take one of the comparator vaccines and go with it. So in very positive term, I'm sure before the end of the year, vaccine will be in the market.
Okay, good. Thank you. All the best.
Thank you.
Okay. So, sir, there is one question from the Shashank Tripathi. Your views on studies showing integration of viral RNA fragment into cellular genomes and its implication for mRNA vaccine?
Not possible at all. Recent, I think last week, a PNS paper has come. It only reached up to the cytoplasm, so there is no possibility of integration into the human genome system.
Okay, so there is no more questions, sir. So okay, so thank you so much, sir, for your time, and it's very informative and very nice presentation. So thank you so much, sir.
Thank you very much. The best of the rest of the conference today and tomorrow.
Thank you, sir.
And it was pleasure interacting at SGPGI, and thank you very much for the invitation. Bye.
Thanks, sir. Okay, so the next session will be moderated by Dr. Santosh, and I would like to request Dr. Santosh, please go ahead and introduce our next speaker. Thank you.
Yeah, thank you.
Santosh, you are muted.
Is it so? Hello, am I audible?
Yes, now.
Hello? Yeah. So our next speaker is Dr. Shashank Tripathi. He is PhD from International Center for Genetic Engineering and Biotechnology in New Delhi, 2012, and at present working as an assistant professor in Department of Microbiology and Cell Biology, Center for Infectious Disease Research at ISA Bangalore, and he is also a Wellcome Trust DBT Intermediate Fellow. And during his PhD, he has an international research exposure as a visiting research scholar at CDC Atlanta. And post-PhD, he has been trained in a very prestigious institute, School of Medicine, with very renowned virologist, Professor Adolf Garcia Sastre. If my apology if I may have not taken a correct name. And then he has been still continued there in the same group as an assistant research professor. And then Dr. Tripathi has been coveted with a number of different awards and fellowship. And as I mentioned, that his lab is supported by the Wellcome Trust DBT India Alliance Intermediate Fellowship 2018. And in addition to that, he's a recipient of the various prestigious awards like the Infosys Young Investigator Award from the Infosys Foundation 2019 and Tata Trust Award Awards in 2019. And Dr. Tripathi has published more than 25 high-impact articles, co-authored and authored in journals like Cell Host and Microbes, Science, Nature Microbiology, Cell. And at present, his group is interested in studying the host interaction of the emerging human RNA viruses, especially focused on the influenza, flaviviruses, and coronaviruses. His group is exploring the broad theme of the virus-host interactions, such as virus invasion, antagonism of the innate immunity, and co-option of the cellular machineries, which can be targeted to develop the broad spectrum of the antivirals. For this, his group is using the different omics approaches like a genome-wide CRISPR screen, protein-protein tractome, and a big data analysis. In combination with the conventional virology, reverse genetics approaches, and small animal models, he is trying to combat and study the emerging virus pathogens. They are working towards the development of the novel influenza vaccines, which provide the broad and the long-term protections. For research related to the COVID-2, his group is studying the host interactions to develop the new antiviral strategy. And in addition, he's also exploring the changes in the molecular profile of the COVID-19 patient samples to identify the prognostic markers. So with this brief introduction, I welcome Dr. Tripathi, and over to you, Dr. Shashank Tripathi. Yeah. Thank you.
Thanks a lot, Dr. Santosh. Very kind introduction. I couldn't believe it was about me. Anyways, I'll hope that I can keep the interest level to the same as Dr. Sanjay's talk. That was very impressive. I'll share my screen. When you see, you let me know.
Sure.
You see this?
Yeah, we can.
All right. Okay. So thanks everyone for joining, and thank you the team at SGPGI for inviting me to deliver this talk. Dr. Sanjay covered the most important aspect in trying to combat a pandemic, which is development of vaccines. I'll be talking about different aspects which are sometimes overlooked, but in my view, equally as important if you want to minimize the casualties and try to control the spread of a pandemic virus. We'll talk about immune response to COVID-19 and how studying immune response can give you clues of severity and can give you keys to therapeutic modalities. So to mention briefly, we are at the Center for Infectious Disease Research in ISC. Our lab is called Emerging Viral Pathogens Lab. And I'll start with a disclaimer. So there are a lot of general information in the presentation, but there are a few research data, IP related to that has been protected by Indian National Institute of Science, so filing patents. We are looking for clinical partners to translate some of these, and if anybody in the audience is interested, they can get back to us after the talk. Okay, so nothing new here. We know we are in middle of COVID-19 pandemic, which we are hoping it gets over soon, but it just keeps on dragging, new variants keep on emerging, and the death toll keep on mounting. So as of this morning, there are at least 4 million deaths worldwide which have been caused by this virus. And the virus causative of that pandemic needs no introduction. We know this very well. Pretty much every person who with any sort of scientific, even non-scientific background has become some sort of a virologist now. They understand the virus, that it looks like a crown, it has these spike proteins, and it has a long single-stranded persistent RNA genome inside it. And if you look a little bit more closely, that genome is about 30 kb long, which is quite unusual for an RNA molecule. Obviously, it gets support through coating by nucleoprotein of the viral origin. But from this genome, the virus makes 3 sets of proteins, structural proteins, non-structural proteins, and some accessory proteins. So structural proteins, the one which you hear most about is the spike. But bear in mind that spike is not the only protein. There are at least 28 more proteins which are made by the virus once it infects the cells. And all of them play crucial roles. But especially these accessory proteins, these are not packaged in the virus particle. They don't make the body of the virus, but they play a very crucial role in modulating the host immune response, starting at the innate immune level, going on messing up the adaptive immunity as well. So the COVID-19 disease, which you see, is a result of a lot of monkey business caused by these accessory proteins which are made inside the cell. So the disease management for COVID-19, and for that matter, any other pandemic virus, will have these 4 pillars which are very crucial: diagnosis, prognosis, vaccines, and antivirals. Now, there has been tremendous amount of effort towards developing diagnostic tools, new vaccine candidates, and astonishing progress has been made, which was never seen before in the case of any other disease. New diagnostic kits were developed in span of weeks, similar for vaccines in span of months. Vaccines were developed, they got clinical approvals, and patients started getting the jabs. But the prognosis of the disease and development of antivirals, this has sort of lagged behind. And for disease prognosis, we'll discuss going forward. We are still depending on methods which are not very specific or reliable. And antiviral field has been completely left behind because as compared to vaccine platforms, developing a specific antiviral for a new viral pathogen, it's quite a daunting task. So most of the efforts which we saw were about repurposing the drugs which were already available. Repurposing the drugs which are against other viruses, or repurposing the drugs which are approved for treating other non-infectious conditions in the host. So our talk or our research, as far as this talk goes, will be focused on prognosis and antivirals. So first part, COVID-19 prognosis. So the slide here shows what is the current clinical practice when you want to look at prognosis. Prognosis meaning understanding the progression of the disease in a patient. Now, as the viral infection starts, the host immune response starts mounting. And as the host immune response keeps up to pace, the viral infection should clear out. That is an ideal scenario. But when these two things play out, there are certain phases which the host goes through. And in those stages, say asymptomatic, mild to moderate, severe, or even critical condition of a disease can result. And in these different stages, there are some markers which are currently being looked at in the clinic. Say in the blood, people look for D-dimers or LDH or C-reactive protein. In more severe cases, they look for inflammatory cytokines like IL-6. And other than that, the hypoxia and the chest scan by CT or by X-ray, these are the methods which are being currently used to understand the progression of disease. Now, other than looking at, say, blood oxygen level with small pulse oximeters, pretty much everything requires you to visit a clinic or a diagnostic lab. So, which is quite cumbersome, especially for chest imaging. You need to definitely visit an advanced imaging setup present inside a hospital. So that exposes you to chances of getting nosocomial infection in the hospital setting because multiple patients are visiting those facilities to get tested. So there is a very urgent need of sort of easy to follow prognostic markers for COVID-19. So we decided about looking at this from sort of a deep understanding of the biology or the progression of disease. So this is a very crucial slide here. So what happens once the virus infection is established, say through your nose or your mouth in your upper respiratory tract, virus will infect, say, local epithelial lining of cells. And in these cells, what you get immediately is production of what is called interferons. And they are type 1, 2, or 3 interferons. And these are actually cornerstone of our entire immune response against infectious diseases. So once the interferon is produced in a cell, it will alert the neighboring cell that there is a virus present in the vicinity. So be ready to fight it off. And also they will recruit sentinel cells of immune system and immune effector cell, which will pass on the information to adaptive immunity, both cell and antibody mediated. And that passing over of information happens through a bunch of cytokines which are produced from epithelial cells and later on the immune effector cells, say dendritic and macrophage cells, which have reached the infection site. So there's a cascade effect here. So first the interferons will be produced, then a first set of cytokines will be produced, which will recruit the next set of immune effector cells. Those cells will go on to produce even more cytokines and further interleukins and cytokines and chemokines, which will sort of escalate the immune response. Now, if this immune response is very well regulated, which is shown in yellow, you can get a mild to moderate disease which will fade away and virus replication will stop and infection will be cleared. But remember the accessory proteins which I mentioned, that they mess with your immune response. And other than that, the virus has a lot of other tricks up its sleeve to escape the immunity. And because of that, it keeps on playing this fight with the host immune response and cat and mouse chase game. And it escapes the immune response. And because of that, immune response goes in overdrive and it continues to produce highly inflammatory set of cytokines and chemokines for a very long period of time, which can result into a severe disease. So the severe disease which is seen in case of COVID-19 is not just the result of virus replication at very high titers. Even with a mild replication or a baseline replication of a virus, you can end up with a very severe disease. Severity of disease is primarily contributed by an overdrive immune response of the host itself. Now, we hypothesize that this cascading effect, at some point, it will have cytokines or chemokines or possibly some secretory proteins which are responsible for switching from mild to a severe phenotype. And if you can monitor the expression of those secreted markers, you can understand the progression of disease. And you can actually predict if somebody is going towards a severe disease phenotype. Also, if you can understand this set of molecules which are expressed during progression of disease, some of them may serve as antiviral targets. Either they may be required for virus replication, so you can block them through chemical inhibitors, or they may be responsible for this severe immune response. So you can block them, and that can lead to reduced immunopathology. And actually, some of the drugs which are being used in clinics, say dexamethasone or IL-6 inhibitor antibodies, they are based on that principle of reducing inflammation. So we started with this sort of understanding of the immune response progression in our respiratory tract against the virus. Now, we wanted to find out which are the factors which are produced in the upper respiratory tract as the disease progresses. And for that, multiple studies have already been done, which have used a variety of omics technology platforms to generate data. This slide shows application of omics technologies to understand different aspects of COVID-19 pandemic, not just the soluble factors, but pretty much everything. What genes are involved, their variation between population, variation in their expression, the effect of age, the effect of viral gene diversity on its tropism and the cytopathic effects, as well as the migration of people and effect on the spread of the pandemic. So a lot of omics platforms have been applied to generate a lot of data, but that data has become a problem itself. So we are drowning in data and we're actually thirsting for knowledge. So this needs to be translated. So as they say, data is the new oil. If you can extract, you can get a lot of useful information out of it. And you can see that how this omics data set for, especially for COVID-19, has gone up. So from December 2020, it shows different common set of technologies used to generate omics data set from COVID-19 infected patients or cell culture or animal models, and the overlap between different kind of platforms, the information which has come out. And by April 2020, this has become a huge mesh of data set. So it's ever increasing, but point is this needs to be extracted and so that useful information can come out of it. So we started with this thought that we are going to look at the gene expression profile in the nasopharyngeal samples. An idea being that during different stages of COVID-19 disease progression, some genes will be expressed there, possibly in soluble form, which will be responsible for disease progression to the next stage. And for that, we chose these studies, these 7 studies, which have transcriptomics as well as proteomics data sets. We call them T1 to T4 and P1 to P3, and all of them had nasopharyngeal samples from COVID-19 positive as well as healthy controls. So then we applied this meta-analysis pipeline here. So basically, we had these 7 data sets. So we started filtering by applying these different kind of cutoffs on fold change, on p-values. That gives us a list of genes. And then with those list of genes, we perform an overlap analysis. And we select further down by calculating cumulative score. And in the end, we do experimental verification of these genes that they are really upregulated in COVID-19 patients. And that has helped us in identifying prognostic markers as well as therapeutic targets. So this whole pipeline, we go through one by one as we move forward. So this is a simple overlap analysis of the number of genes coming across from different data sets, T1 to T4 and T1 to T3. Each box shows the overlap coming between, say, these two different kind of data. And these are the p-values, which represent what is the sort of confidence in overlap between two different data sets. Long story short, if you combine all of it, you get a number of genes, which is about 566. Let me try to hide it. All right, so those 566 genes, if you combine them and look at what kind of biological functions they are involved in. So as we had predicted that we are looking at things which are produced in the nasal cavity or upper respiratory tract, and most likely they are going to be involved in immune modulation. And that's what we find actually. So most prominent, some of the most prominent biological pathways they are involved in are type 2 interferon signaling and response to interferon alpha, interferon gamma, as such. And when you look at how many of these genes are regulated by interferon, this can be done by probing these gene data sets in interferon database. We find that pretty much all of them are regulated by type 1 and type 2 interferon, which again sort of validates with our hypothesis in the beginning. Now, we further reasoned that anything which is supported both by protein overexpression as well as RNA overexpression is likely to be true, which is called support from orthogonal data sets. Because in the end, the phenotypic changes will come about mostly, sometimes it can come about by RNA, but mostly it will come about by proteins. So then we shortlisted things which are supported by both protein as well as transcription data sets. So when you compare protein transcription versus protein and transcription versus transcription data sets, you get this kind of Venn diagram. One of the factors which comes in the center of it, which is supported by all data sets, is thyroidoxin. We'll come to it at the end of the talk. But when you shortlist these things, say, which are shown here, 1, 2, and 2, which are present in both protein data sets, and shortlist these green circle things, which are 16, 26, 1, and 1, which are present in transcript and protein data sets. Then combining this will give you a list of genes, which are about 46, and each gene is supported both by transcription as well as protein upregulation. Now, if you look at their biological function, pretty much everything is exclusively related to immune response. So this gave us a little bit more confidence that we have narrowed down the list further towards the desired phenotype. And still everything is still regulated by interferon, as was seen earlier. Now, these 46 genes are listed here, and their individual expression level in different data sets is shown here. Red meaning more overexpressed in a particular study. And then we have calculated a cumulative score for each gene across different study, and that cumulative score ranking is shown here. So this is a list of genes which are overexpressed both at protein and RNA level in the nasopharyngeal swabs, as identified through meta-analysis of a large set of omics data. So these are the potential genes which can serve as prognostic markers. That was our hypothesis. So obviously 46 genes were quite a bit for us to screen. So we shortlisted further on. One of the family of genes which caught our interest was S100 family, because there were a couple of them in the top, and there are more S100s in the subsequently down in the list of genes. So we took all the S100s and top 10 from here. We also took thyroidoxin because it was in the center of our overlap diagram supported by all the studies. And then we went on to validate overexpression of these genes in a cohort of COVID-19 patients. So we took COVID-19 samples from COVID-19 diagnostic laboratory, which is set up in CIDR, and we divided them in 4 categories, either COVID negative or COVID positive. And within negative and positive, either they were asymptomatic and symptomatic. And when we say positive symptomatic, that means really severe symptoms of COVID-19. And their age distribution and their gender distribution is shown here. Then we went on to validate those shortlisted genes by RT-PCR, whether or not they were really overexpressed in the patient cohort, which was present in our data, which is present in our facility. Remember, the meta-analysis came about from using data sets from different geographical locations, some in China, some in Europe, some in USA. And now this is validation of that in an Indian cohort. So it holds true in different geographical locations as well, that all the S100s in positive symptomatic patients are highly upregulated. Pretty much everything is highly upregulated. The black bars show that the gene was not detected in a particular patient. And at the bottom here, you see the expression level of, say, viral RNA. That correlates very well, that in positive symptomatic patients, the viral RNA level was high as well, although we chose the samples which had low CT values. So if you try to calculate the significance of these genes when you try to compare between different categories, you get this kind of diagram. I'll explain this. So if you compare negative asymptomatic versus positive symptomatic, this becomes an infection marker or sort of a diagnostic marker. When you compare negative symptomatic or positive symptomatic, this also becomes a COVID-19 marker, infection marker, but more specific for COVID-19. And when you compare positive asymptomatic versus positive symptomatic, meaning COVID-19 positive, but either without any symptom or with very severe symptoms, this is the kind of comparison which will give you severity marker or a prognosis marker. And those comparisons are shown here. The first is infection marker, the second column is COVID-19 marker, and third column is the prognostic marker. And you can see the performance of all these genes in different kind of comparisons. So you can see that for the prognosis, most of these genes work very well, especially the S100 families. They are very significant. So then we went on to do what is called ROC curve analysis, or receiver operator characteristic analysis, which is usually done to identify diagnostic or prognostic marker. And basically here you plot the sensitivity and 1 minus specificity on a plot like that. And you use the CT value for every patient in your cohort. And if this curve moves towards the top here, that's where you can call it a very highly sensitive and specific diagnostic or prognostic marker. And if the area under curve is anywhere above 75, you can directly say that it is 75% sensitive and whatever the corresponding specificity is. So for all the genes shortlisted, here are the area under the curve. And especially if you look at the S100s, they have pretty high area under the curve, among the highest in this gene set. If you look individually for the S100s in our study, then what are the corresponding sensitivity and specificity when you're trying to differentiate between, say, negative, positive asymptomatic versus positive symptomatic. You can see that S100A can identify severe symptoms by 93% sensitivity and 68% specificity, and others also behave not too bad. So this is a signature we identified, which can very reliably identify the disease progression of the COVID-19 using, keep in mind, the nasopharyngeal swab samples. So what are these genes, S100s? So these are secreted immunomodulators, the same kind of phenotype which we're looking from when we started the study. And they act through what is called RAGE signaling, and they activate NF-κB and AKP1 signaling, which further lead to production of more immunomodulatory chemokines and cytokines. And more detailed diagram is shown here. So they will produce different interleukins, which will recruit more cells. And basically, we were looking for these molecules which may serve as a bridge between innate and adaptive immunity, and sometimes may be responsible for severe disease. And these S100s are precisely that. So the advantage of this signature here is that these can be measured by simple RT-PCR in the nasopharyngeal swab samples, which are routinely collected for RT-PCR based diagnosis. Meaning the same sample which is collected for RT-PCR diagnosis, in the same sample you can look for S100 signature, and you can also get sort of an estimate whether or not this case is moving towards a severe disease, which will obviate the need for really doing blood-based markers, which are not really COVID-19 specific, but are organ damage markers, and possibly prepare the healthcare workers better in allotting resources and preparing for patients which may be moving towards a severe disease phenotype. So that was the part about prognosis. Moving on to the antivirals and therapeutics part. So as I mentioned in the beginning, that this is a part which has lagged behind, mostly because of the technical challenges and the regulatory processes, which are much more complicated here. Usually, there are only a handful of antivirals which are available in clinic for just under 4 or 5 different viruses, and all of them have come about after decades of research. So compared to vaccine development, antiviral development is much more tedious and expensive as well. However, the options people looked at is host-directed therapy, as well as the repurposing of antivirals which were developed against other viruses, say Ebola, SARS-1, MERS, or even influenza. So this diagram here shows the replication cycle of SARS-CoV-2 and different steps of it, which can be sort of blocked or targeted through different kind of molecules which are available, either in clinic or in some experimental stage. So the virus enters through engagement of ACE2 receptors or TMPRSS2, and then once inside, it will deliver its RNA genome, which needs to be transcribed and replicated. So entry of the virus can be blocked by chloroquine, chemostat, or the recombinant human ACE2, or even antibodies against spike. And actually, the vaccines also would work using the same principle. They will primarily work by neutralization, which is inhibition of the binding of spike to its receptor and thus preventing entry. And then subsequently, this transcription replication step can be blocked by nucleoside analogs, say remdesivir, ribavirin, and favipiravir. And then there are proteases in this virus which can be inhibited. But remember, all these proteases inhibitors, lopinavir, ritonavir, or the nucleoside analogs, these are repurposed. There's nothing which has been developed for SARS-CoV-2, and I don't see it being developed that quickly. It will take a lot of time. Although multiple papers have come out which have very quickly identified protease inhibitors and new set of nucleoside analogs, but it will be some time before they pass through the clinical trial pipeline. And moving on forward, the virus will assemble and come out of the cell, and it will go on and infect next stage of cells. So there are some inhibitors which will block, say, virus egress also. But the other important aspect is when the virus is out and about infecting and killing cells, it will raise alarm, which will recruit a lot of host immune effector cells, and they will produce inflammatory cytokines. So there are host-directed therapies as well. Say there's IL-6 production is there, so there are IL-6 anti-IL-6 receptor monoclonal antibodies, or there's dexamethasone which is being used to reduce the inflammation. So these are the options which are available. Now, as I said, if you understand the set of reactions going on here in the upper respiratory tract where different molecules are being produced, as well as if you can understand what is going on inside the cell where the virus is replicating, some of these host factors may serve as targets for therapy. And those host factors might as well be present in the gene set which we identified in our meta-analysis. And one of those, as I mentioned earlier, was thyrodoxin. So thyrodoxin, surprisingly, was not very highly upregulated. So hence, it could not serve as a very reliable prognostic marker. But it was upregulated mildly, but very consistently in pretty much every data set we looked at. So hence, we decided to look at it in more deeper. Now, what is this protein? So this is part of this thyrodoxin system where it flips between oxidized and reduced state, basically disulfide bond form or removed. And this reaction happens through an enzyme called thyrodoxin reductase. And through this reaction, it can transfer disulfide bond or facilitate disulfide bond formation on multiple target proteins, and through which it can regulate different kind of cellular processes. What cellular process it regulates? Reactive oxygen species production, so redox responses, which are directly involved in inflammation, cell death, and also ubiquitin proteasome system. And its direct targets are NF-kappa B, AMPK, P1, transcription factors, all of which are implicated in production of inflammatory cytokines. Now, as I mentioned, that thyrodoxin from oxidized to reduced state converted by this enzyme called thyrodoxin reductase, and there is a FDA-approved drug called auranofin. It's a gold drug which can block this reaction by binding to thyrodoxin reductase. This drug, as you would expect, is used to inhibit inflammation. Now, this is used for treatment of rheumatoid arthritis, has been around for 1985, but this is not the first drug which is preferred for treating rheumatoid arthritis because it has some side effect in long term, especially diarrhea, because arthritis patients require anti-inflammatory molecules to be taken for a long period of time. So this is not used as a first line of treatment, but anyways, this is an approved drug for this condition. And in some studies, at least in cell culture-based studies, it has shown to be effective against multiple infectious agents such as HIV and TB as well. Even in case of ovarian cancer, it has been tried. This drug is also available in India, manufactured by Cadila, and the market name is Goldar. This comes about 115 rupees for 10 capsules. Pretty safe to use, already approved, have been around for a long time. And it inhibits this thyroidexin reductase enzyme, which will prevent whatever function thyroidexin is supposed to do inside the cell. So then we decided to test whether this drug will have any effect on virus replication. Hypothesis being this thyroidexin system is consistently being upregulated in virus-infected cell or surrounding. So it may have some role to play either in virus replication itself or in modulating the subsequent immune response. So we started with testing the toxicity of this drug in cell culture, HEK-ACE2 cells, and we found that this is sort of tolerated up to 2 micromolar dose. So that's what we decided to use moving on forward. And then we tested its effect on viral replication in some cells in reducing amount. And at 1 micromolar, you can see that about 2.5 log reduction was observed. And you can see by Western blot, the spike protein in this drug-treated condition is gone completely. And the same was validated by plaque assay as well. And this effective concentration comes to about 0.29 micromolar, which corresponds to the dose which is clinically used for auranofin for treatment of arthritis. We went on further to test whether it is reducing the cytopathic effect caused by the virus in cells, and you can see that, say, if this is the cell control and virus leads to reduction in viability by 90%. In case of auranofin treated cells, the viability is rescued. That can be seen visibly as well. Here you see a lot of CP in virus control, and upon drug treatment, it starts to look similar to what you would see otherwise in uninfected control. We didn't stop here. We went on to further validate the efficacy of this drug in a preclinical Syrian hamster challenge model. And there we tested the drug in both prophylactic as well as therapeutic setting. So prophylactic meaning the hamsters were provided drug on consecutive days for 3 days and then challenged with the virus. And in therapeutic setting, they were provided drug for 3 consecutive days while the infection was on. And on day 4, the animals were sacrificed to look at the status of the viral infection titer and the disease progression. And here I'm showing you the toxicity of this drug in hamsters. Even up to 5 mg per kg, there was no effect on animal health. They could tolerate it very well. And then when given in case of infection scenario, you can see this therapeutic and the prophylactic drug treated group of animals, they lose very little weight or recover pretty quickly compared to an infected group of animals which lost more body weight. And this corresponds to reduction in virus titer as well. Compared to vehicle control and infected, drug treated group, both prophylactic and therapeutic, had reduced virus titer as measured by RT-PCR of the lungs, reduced by almost a log. And same is reflected in the health of the lung as seen by histopathology. So in case of infection control, you see a lot of tissue damage, infiltration of cells, edema, loss of epithelial linings in the alveoli, loss of alveolar structure. But in case of healthy control as well as drug-treated control, the structure of alveoli is maintained and the lung health is maintained. Potential mechanism of action. So as we had seen that thyroid is upregulated in patient samples, we validated whether or not it is upregulated in the animal model. And as one expect that in infected groups, it was always upregulated. But one hypothesis was that thyroxine works by inhibiting inflammatory response. So we looked at the most important inflammation mediator, IL-6, And as you can see, IL-6 goes up compared to a healthy group upon infection, but when you have drug present in the animals upon infection, the IL-6 production is blocked. So in principle, this drug does the same function which is done by, say, tocilizumab, which is inhibiting IL-6 function. And if you compare the cost, this is almost more than 100,000 times cheaper than tocilizumab monoclonal antibodies. So this is summary of the whole study here. We started with meta-analysis of the omics data, especially from the nasopharyngeal swabs, and we identified a gene signature which can be, simply tested by RT-PCR in the same set of samples which are otherwise collected for diagnosis of COVID-19. And moving on further, we validated this signature in a cohort of patients, and we calculated the sensitivity and specificity, which are at par with any other disease signature which is being used for other conditions. And also we identified this drug auranofin, which can block thyroidopsin, and it can result to reduce virus replication as well as reduce inflammation, which leads to disease severity. So this work has been done by amazing students in my lab, Abhijit and Wahida. They led the work primarily, supported by Shachi and Dr. Rohan Narayan, who's handling all the BSL3 work, especially with the hamster model. And this has been also supported by the COVID-19 diagnostic lab here, which is run by Professor Amit Singh and also Professor Deepak Sani. We got some help from Professor Dinesh Gupta in ICGB with the bioinformatic analysis part. And all the animal challenge studies as well as live virus infections were done in BSL3 facility. Our lab is supported by startup funds from ISC, Young Institute Award from Infosys Foundation, DBT BIRAC, as well as Welcome Trust India Alliance, and all their support is acknowledged here, which has allowed us to conduct this work. So that's all I have, and I'll take questions.
So thank you, Dr. Tripathi, for such an illuminating talk. And I think it would be going to be very beneficial for the whole audience. So maybe I can start to have a question and session. So may begin from my end itself. So I have a question, say, related to the your first part of the study with the COVID prognosis. So I think in some part of your slide, you mentioned that you have taken a nasopharyngeal swab as well as the BALF sample as well. Yeah. So do you see this signature, say, the secretory proteins? Because in the early phase of, say, infection, these ascended proteins, is it cell type specific, or immune cells are also going to be involved in, say, the secretion of cells? And how are you going to correlate in terms of, say, if you...
Tosh, I lost your audio. I'm sorry, I'm unable to hear you, Santosh.
Can you hear me?
Yes. You're muted again. Something's happening.
Yeah, muted. Let me ask you. Hello?
Yeah, I hear you now.
Hello? Hello?
Yes, I can hear you, Santosh.
Yeah. There was some sort of a, say, network issue in our... So have you got my question?
No. Can you repeat, please?
Yeah, means in terms of, say, your first part of the talk, you mentioned that you have taken somewhere the sample, the nasopharyngeal swab, as well as the BALF sample. So my concern is, say, at the early time point of the infection at the upper respiratory tract, and at the BALF sample, which are going to be the deep inside the lung. So how this, is there any kind of a difference in, say, the expression level, or as well, the secretion level of these proteins at the same time point? Because the virus traverse means at the early phase of an infection, the secretion of these S100 proteins, and possibly at the in the bulk sample may be somewhat different and along with that, like say the level of like say the virus infected cells and like say the contribution of these S100 cells to other immune cells, how they may help like say in to track the disease progression.
Yeah, so since our data analysis initially considered studies which had these gene expression profiles combined from both nasal swab and BARF, so we cannot really differentiate that. Also, the samples which are collected for diagnosis, at least in our facility, they usually have these two swabs they take, one from the nose and one deep down in your throat as well. So then both sticks are put together in the BTM for collection of whatever cells are there in that swab. So I would assume that it should cover partially whatever you expect to see in BALF. But in nasal swab, definitely everything will be covered. But I cannot differentiate between, say, very high up in your nasal tract and then at different stages of your respiratory tract, how the progression of the virus or the expression of host genes will vary. But I would assume that virus will move pretty fast. And the prognostic markers which we are talking about, they will come into play or they will express at high levels at least at few days later on when the virus infection has prevailed. Very early on, what you'll get is like interferon production.
Hello?
Yeah.
There's some. So, and then my second question is in terms of like the sensitivity of this assay, because we do consider like say the RT-PCR is a gold standard, but how this, what is means secretory S100 proteins, the assay which you possibly may going to develop. So in terms of like say how to correlate with like say the RT-PCR positive samples and the extent of like say the expression. And to add my point, like say means in the second wave we have seen like that patients who are RT-PCR negative, but how they are going to correlate with the secretion of the such S100 secretory proteins?
No, so patient testing RT-PCR negative in second wave is a different issue. That's a diagnosis issue that primers were not updated and possibly there was a mismatch and because of that you're not picking them up. But for as far as our plan is, we'll stick with RT-PCR assay only so that the same infrastructure setup and set of people who are trained in that technique, they can do both COVID-19 diagnosis as well as detection of the prognostic signature. Now, our study right now, whatever you see right now is limited to a small cohort where we have distinction either asymptomatic or symptomatic. What we're planning to do now is to go with a much larger cohort where we'll have more fine divisions, say completely asymptomatic or required a hospital visit, or there was hypoxia, or there was an ICU admission, or there was ventilation or ECMO support, or somebody deceased eventually. Or in between, you can throw in say vaccinated or not vaccinated, first or second dose vaccinated, or which vaccine candidate they got, primary infection versus a secondary infection, things like that. So we need to validate this signature in a much larger cohort with finer categorization. And but the strategy will remain the same. It has to be done by RT-PCR only. For the sake of simplicity, and that's the whole advantage here, that you don't need to draw blood, you don't need to extract. Anyways, collecting nasal swab itself, a lot of people consider that very uncomfortable. So why not spare them from any more misery, and just that sample can be repurposed for giving them information on potential outcome of the infection.
So and my question is because this S100 protein you mentioned like say they are like say the calcium dependent. So it's all like say the viruses which are going to be infected. What's the upstream modulator which makes to secrete these S100 protein, or is it both in like say the infected and if possible that how this calcium signaling are going to modulate the microenvironment of the immune cells to secrete, because that may, I think, like say vary to extent of the proteins.
No, so that's a general question about S100 biology itself. I mean, I don't think their mode of action is going to change between different kind of infections. And I also don't think that they really help by any means in virus replication or completing its replication cycle. Their primary function is very well established, that they go through this RAGE signaling pathway and stimulate inflammatory cytokine production, mostly in immune effector cells, macrophages and such. And that mechanism is going to remain the same. I should also mention that we are not claiming that this is a COVID-19 diagnostic marker. So S100s are reported to be upregulated in other inflammatory and infectious conditions as well. But when you look within the COVID-19 positive cohort, then it can definitely tell you about the progression of the disease.
Thank you. So then I will have like say the questions from my colleague, Dr. Alok as well.
Very nice presentation, Dr. Sasan, and it's very interesting and very important points, important issues about the cytokine storm or involvement of these key markers like S100 and all these stuff. So my question is that, as you said that you are using the BSL-3 facilities for these kind of studies. So are you using any kind of the knockout or any kind of available in vivo model for these markers, means S100 or some kind of pathomechanism determinant?
No, I mean, as far as in vivo studies go, we are sticking with the hamster model now, because they're continuing to own it a bit more, learn more about it, develop new reagents. But whether or not S100 expression is there in the hamster model itself, we have not tested that yet. That's one of the things we'll do. And does it correlate with the disease progression? Does it correlate with the viral load? Things like that. Those studies are ongoing.
Okay.
So,
okay, may I ask?
Yeah. Our we have a question from our Dr. Loken.
Sure.
Yeah, so Dr. Tripathi, it was a very good piece of work. I have just one concern, that since you mentioned this S100 again, because that's what you were presented. So S100 is, I think it's a general response marker in terms of stress marker rather, because it is also in the clinical practice, like in terms of traumatic brain injury and various cancers. Also in these markers are being used as a serving as a prognosis. So I'm just wondering, in those patients where you have collected the samples, have you followed them up? Because and nowadays in COVID, that's what we are learning, that post COVID complications, one of the complications include the brain disorders, or maybe some sort of stroke also sometimes people have reported. So have you looked at, or have you followed up those patients, like how they recovered, and after recovery do they have any sort of problems? Because that can be a more clear idea if that is to be used as a prognosis for certain diseases in terms of COVID.
Yeah, I mean, certainly we would like to do that. We have not done that yet. And as I said, the data which I showed you as of today is based on that small cohort, which is a retrospective cohort. We already knew the patients and their COVID results. What we need to do is a prospective cohort, which ideally should contain the component for follow-up. But I mean, somebody in ICU or somebody diseased, definitely they cannot answer your phone call. But
but I think
ideally we should do that. But precisely that's what I mentioned, that this is something which can be done better in a clinical diagnostic setting, diagnostic lab which is set up inside a hospital, so that you can recruit patients prospectively. Our limitation is that we are just a diagnostic lab in an academic institution. We don't have direct interface with the patients. So retrospectively, we did whatever we could, but moving on forward, we need a clinical partner to do a much larger scale, what you can call a randomized phase 1 trial.
Yeah, but I think I just a suggestion, because your sample size is not too big, so it won't be too much difficult to follow those patients, because at least you may have some information about those patients, some sort of their contact number, or because that's what nowadays government is also focusing on, tracing, or maybe kind of
Certainly.
following up with the patient even after vaccination or something else, since you already have some leads in terms of S100 or thyroidoxin, which again gives a question, because these are general response proteins which are again activated in terms of some sort of stress or response as an inflammation. So I think, for sum up to your work, I think it will be really very interesting to see if it can be correlated with some sort of post COVID complications, and if those patients, because since you mentioned that you all only measured these parameters in swab samples or nasopharyngeal samples, so I don't know in blood samples, because generally in clinical practice serum samples or blood samples are used to look at these parameters. So I don't know how much, since already in saliva or in these kind of samples, if it is there, definitely in the serum it will be much higher. So I think it will be a really interesting aspect to look at. So anyway.
No, sure. All points well taken, and we plan to do that. And the idea here is two things to clarify again, that these are general stress marker, which is correct. They are upregulated in other infection or damage situation also. But we are looking at only COVID positive patient samples, and within that, they can inform you about prognosis. That's what the claim here is. And definitely, with a much larger cohort and follow up, we'll learn more about how these things express, how the signature holds. We will do that.
Okay, thank you.
All right. Santosh, you are muted again.
Hello. Yeah. Let's see, like, if Professor Swasti is also around, and if she has any question. Ma'am? No. I don't see also any further questions in the chat box. So with this, I would like to thank Dr. Tripathi for such a illuminating and exciting talk. And I think it was very helpful for all the audience and for all of us. So, and we look for in future further potential further discussion related to COVID. And thanks for your time and presentation as well.
Thank you.
Thank you very much.
Thanks, Alok. Thanks, everybody at SGPG. Thank you.
Okay. So with this, we would like to end up our today's session, and we will further look forward to have like for the next day session tomorrow at 10 a.m. Thank you. Thank you very much.